In this module, we look at an animation of PCR reaction. So let's look at that. Okay, let's look at our PCR animation. Here's our DNA. As I mentioned, that the first step in PCR is denaturing the DNA, which means separating the two DNA strands. We are going to break the hydrogen bonds. In our cell, the same function is done by helicase enzyme. Here in the machine, PCR machine, as I said, is basically a fancy machine that regulates the temperature of the sample. So first we are going to raise the temperature to 94 to 95 degrees, and when that happens, it allows the two DNA strands to separate and reveal the information, which is basically the sequence of nucleotides. In next stage, the temperature of the sample is lowered to 50 to 65 degrees Celsius. This is the temperature which allows the primers to bind to their specific or complementary sites in the DNA, on the DNA molecule. So now we are going to anneal the primers or make them hybridize with the DNA. Here are our primers. So these primers, they are complementary to the specific DNA nucleotide sequence which is present on our target DNA. So both these primers are going to be extended when we raise the temperature to 72 degrees Celsius. This temperature is ideal for the working of this enzyme. It is most efficient at this temperature. I should mention that this enzyme was isolated from a bacteria that grows at very high temperature. The name of the bacteria, of course, most people are familiar with, is Thermus aquaticus. So we raise the temperature, the enzyme becomes active. Here is our Taq DNA polymerase. The primers are extended. Here, the 3' direction, in the 5' to 3' direction, we have extended our primers, and primers have been incorporated into the new DNA molecule. This is the region of interest. This is the part of DNA that we want to make many copies of. In the next stage, we will again repeat the cycle. We go to the second cycle. We denature the DNA. We again lower the temperature, 50 to 65 degrees Celsius. The primers anneal. We extend the primers, and here the primers have been extended. Please note, in two PCR reactions, we have gone from 1 DNA strand to 4 DNA strands, and however, thus far we haven't specifically isolated, we haven't specifically amplified or made many copies of just the fragment of DNA that we want to. This is cycle number 2. We have gone from 1 DNA to 4 DNA strands. Let's go to the third cycle. When we go to the third cycle, again we raise the temperature to 94 degrees Celsius. All the DNA strands separate. They are no longer in a form of double helix. We lower the temperature. The primers bind their specific complementary sites on the DNA molecules. We again raise the temperature, and the primers have been extended, and now we have 8 DNA molecules. This is exponential growth of DNA, amplification of DNA. Note that in cycle number 3, this is the first time that we have specifically made the target DNA. We now have a piece of DNA that we were interested in amplifying. So this is cycle number 3. We have 8 copies out of 8. The 2 copies of DNA are the ones that we are interested in. That is our region of interest. We go to the 4th cycle. We repeat the same procedure. Now we have, in 4th cycle, we have 8 molecules of DNA which are the right size, which is the right fragment that we wanted to amplify. Go to the 5th cycle. It keeps on increasing. The amount of DNA keeps on increasing exponentially. Let's look at the graph. Remember, cycle number 1, we had not made any copies. After cycle number 1, we had made 2 copies of DNA. From 1, we had gone to 2. In cycle number 2, we had made 4 copies of DNA. Remember, there is no target DNA copies present. In second cycle, it appears for the first time in the third cycle, and we have 2. So let's go to the next cycle. Cycle number 3, remember, we had 2 target copies and a total of 8 DNA molecules. In 4th cycle, we had 16 total copies of DNA and 8 target copies. Let's keep on going. So here we have our PCR reaction going on. We are 16, 17, 19, 20, and let's run it to 30 cycles. And after 30 cycles, we have more than 1 billion copies of the target DNA. It's a simple technique which has allowed us to go from one molecule of DNA, part of the same molecule has been amplified more than 1 billion copies of that particular fragment of DNA. So this is the power of this technique. Even if your sample contains very little amount of DNA, you can amplify it and detect for the presence of that particular DNA. How can this technology be used for human health and improving human society? We will look at that aspect in the subsequent modules. I hope you enjoyed the animation.